So our next speaker is Professor Shyh-Dar Li from University of British Columbia. So let me introduce Professor Li to the audience. So Dr. Li has received his BSc in Pharmacy from National Taiwan University in 1998 and Ph.D. in Pharmaceutical Sciences from the University of North Carolina at Chapel Hill in 2008. He finished his postdoctoral training at Morris Cancer Center at the University of California, San Diego in 2009. He is now an associate professor at UBC Pharmaceutical Sciences. Dr. Li has won several research awards, including 2014 AFPC New Investigator Award, 2013 AAPS New Investigator Award in Pharmaceutics and Pharmaceutical Technologies, 2013 CIHR New Investigator Award, 2013 CSPS Early Career Award, and many more. He is the chair-elect of the Targeted Drug Delivery and Prodrug Focus Group at AAPS and an academic editor of BioMed Research International. Dr. Li's research focuses on developing innovative drug delivery systems to enhance cancer chemotherapy. His research program has been well supported by the NIH and CIHR. So with this, I invite Professor Li for the talk. Now it's over to you, Professor Li.
Thanks so much for the kind introduction and the invitation for me to share my work with you today. So today I'm just going to share a little bit about my research program at University of British Columbia in Canada. So at UBC, we are working on drug delivery technologies for targeted therapies. And we are working on many different technologies, including nanoparticles and prodrugs. And today I just want to talk about one of our focuses, which is the liposomes. So many of you are very familiar with liposomes. So the structure of liposomes is shown here. Basically, is a as you can see, this is a liposome. The structure is basically a lipid bilayer. And for drug delivery purposes, we actually make these liposomes in the nano size. So you can view liposomes as a nano-sized cell. So it's able to deliver both hydrophobic and hydrophilic drugs. But in our lab, we are mainly focused on fully water-soluble drugs. And today I want to discuss two research topics we are working on with liposomes. The first one is that how do we change lipid composition or the fabrication way to make liposomes targeted to different disease sites. And today I'm going to focus on hepatocytes. And second part, as I mentioned, we are interested in targeted delivery of fully water-soluble drugs. But what we are working on is actually developing a very unique active loading technology to load those fully water-soluble drugs into the aqueous core of liposomes, so that we are enhancing the stability of the drug during the circulation and also using nanoparticle lipid nanoparticles, in this case liposomes, to target these drugs to the disease sites. Again, many of you are familiar with the manufacturing of liposomes. So it all started with a thin lipid film, and with hydration, then the lipids will self-assemble into so-called multilamellar vesicles. And we can further extrude MLV into SUV, stands for small unilamellar vesicles. And by just passing them through a very precise membrane with precise pore size. So this is how we create SUV or liposomes for a targeted drug delivery. And in terms of drug loading, right now most people are using the so-called active loading drug technology. So first they create a transmembrane gradient that will allow drug to participate into the inner aqueous core. But definitely there are some limitations for this thin film hydration method and active targeting method. And this is what our lab is currently working on to address those limitations. First, I want to talk about a new project in the lab. Is phospholipid-free small unit lamellar vesicle, or so-called PFSUV. So, as I mentioned, for liposome or liposomal SUV, they are composed of phospholipids. But we also know that SUVs can form with just cholesterol and non-ionic surfactant at 1:1 molar ratio. But this standard so-called neosome formulation is actually not very stable in the plasma because it has to contain a very high non-ionic surfactant component in the formulation to help cholesterol disperse in this good lipid bilayer structure. So that creates a lot of problem because of poor stability. Then the formulation is mainly used for topical application, not for systemic application. So here at UBC, we are using this very innovative microfluidic system called herringbone microfluidics. So it allows organic solvents to collide with aqueous buffer in a very efficient and very rapid way. So in this case, we can dissolve lipids or other poorly water-soluble components in an organic solvent and mix with an aqueous buffer within this microfluidics to produce our nanoparticles. So in this case, we actually just used cholesterol and twin-80, which is one of the non-ionic surfactants that are commonly used for drug delivery. So our goal was to increase cholesterol content as high as possible, and we were able to achieve a 5:1 molar ratio of cholesterol to twin-80. So this, as you can see, with this technology or fabrication method, we were able to produce SUV contains no phospholipids, but only cholesterol and twin-80 in a very efficient way. And these SUVs, as you can see, they contain a very nice lipid bilayer, and the size is about 50 to 60 nanometer up to 18 nanometer. So pretty small. And we were wondering whether these nanoparticles can be loaded with drugs using active loading technology. So we use a weak-based drug as a model drug. And so as you can see that when we create this pH gradient, we were able to drive this weak-based drug, which is doxorubicin, into the aqueous core of the PFSUVs. So these doxorubicin actually form crystals inside the SUVs. You can see this nice structure inside. The next thing we tested is the stability of PFSUVs in plasma. As I mentioned earlier, the standard neosome formulation, because they contain a lot of non-ionic surfactant, that makes them very unstable in plasma. But in this case, we look at the drug retention in the serum and then compare two different formulations. One is PFSUV, the other is pegylated liposomal doxorubicin, which is the equivalent of Doxil or Calyx, currently used in the clinic for breast cancer therapy. And you can see that they are almost as stable. You see that within 3 days, there's no drug leakage from PFSUV in the presence of serum. And even at day 6, we still maintain 90% of the drug content inside the PFSUV. So quite stable. Then this makes it feasible for this formulation to be administered through a systemic route. So this is a in vivo imaging study. We label PFSUVs with a fluorescent dye and then injected these fluorescently labeled PFSUVs into mice. And over time, we just image those mice. And it's not really surprising to see a very rapid accumulation in the liver. But I have to say that the tissue targeting is pretty specific to the liver. And what is really interesting is that when we look into the liver section, and we discover that these nanoparticles are actually targeting the hepatocytes rather than the Kupffer cells, like most of other nanoparticles reported in the literature. So in here, this is a liver section, and we scan the outline, the skeleton of the cells with phalloidin, so you can see the cell outline pretty well in green, and our nanoparticles are labeled in red. And with this, you can clearly differentiate the Kupffer cells from hepatocytes by the green outline and the nuclear morphology. So for example, this is a Kupffer cell, but this big structure is a hepatocyte. And you can pretty much appreciate that most of the red signal are within the hepatocytes. And you see some of the Kupffer cells have some uptake, but for example, this one doesn't really have any red signal. But in here, most of the hepatocytes are stained red. So it's suggesting very selective targeting for hepatocytes for our PFSUVs. So we did some quantitative analysis looking at DIR, which is the fluorescent dye positive cells per field, and you see hepatocyte or the major cell type. On the other hand, we also look at within each cell type, do we have a higher uptake? As you can see here, only among all the hepatocytes, 70% of them are stained with PFSUVs, but only 30% of the Kupffer cells are positive with our PFSUVs, again suggesting this is a really hepatocyte selected delivery system. So we have performed some preliminary study to look at the mechanism of this PFSUV for hepatocyte targeting. And our preliminary data suggested that these nanoparticles actually, when they are injected into the blood circulation, they absorb apolipoproteins to their surface. And because of the small size, they were able to penetrate through the liver fenestra and then into the space that can interact with the hepatocytes through the LDL receptor pathway. And the following just that we did some work just to show that these very unique PFSUV particles are able to target drugs to have some medical applications. So in this case, we are looking at acetaminophen induced liver injury. So this is still the #1 cause for acute liver injury in the Western world. So the model is induced by IP injection of 500 milligram per kilogram APAP, which is acetaminophen. And 1 hour later, we injected a drug called chlorpromazine, which is an autophagy inducer that has been shown to rescue liver or hepatocytes after acetaminophen acute tox. And 6 hours later, we collect the plasma and also the liver to examine the liver toxicity. So as you can see here, the untreated liver shows a big region of necrosis. With some free drug treatment, you were able to see reduction of the necrotic area, but the necrosis is still significant. With our PFSUV, the liver histology looks almost normal. And we also compare ALT, which is the liver enzyme that is reduced after liver is injured. Again, we can see that with PFSUV treatment, the ALT, serum ALT concentration is much reduced compared to untreated control and free drug. So the other application we look at is the liver stage malaria. So malaria is still the most prevalent parasitic diseases in the world. So it causes a lot of death each year. We do have a lot of drugs that are very effective against the circulating parasites of malaria. But the problem is those bugs actually hide in the liver. And so those drugs are no longer effective against the liver stage malaria. And those parasites can relapse anytime. So it becomes very difficult to cure malaria in this case. But we do have at least one drug that is effective for both circulating and liver stage malaria. One of them is primaquine. The structure is shown here is a wheat-based drug. The problem is that this drug is inducing very significant hemolysis. So this significant toxicity prevents its wide use in older patients. So we were wondering maybe our PFSUV can target this primaquine, this PQ drug, to the hepatocytes more effectively. But in the meantime, during the circulation, the particles were able to protect the drug from entering into the RBCs to reduce the hemolysis toxicity. So indeed, when we did the animal study, when we examined the liver uptake, we were able to see a very significantly increased the liver uptake with our PFSUV formulation compared to free PQ by oral or IV. So especially for oral free PQ, our PFSUV increased the liver delivery by 5-fold. On the other hand, the RBC uptake was reduced by 15-fold, so very significant. And we also look at whether PFSUV were able to decrease the RBC toxicity. So this is looking at the RBC morphology after different treatments. So this is a very nice RBC round shape, very nice cell membrane still. But with free PQ treatment, you can see that the membrane is sort of deformed a little bit, suggesting this might be toxicity there. And with the PFSUV, you see the morphology of RBC is preserved better compared to free PQ treatment. So that concludes the part one, which we use a unique fabrication way, and then we clear the lipid composition for a specific cell or tissue targeting. The second part of my talk will be focused on so-called active loading technology. So this is the standard active drug loading technology for liposomes. So as I mentioned, first you create a gradient, a transmembrane gradient between the liposomes, and you just add an antipathic drug in the external media. And because the drug is soluble, so it will be soluble in the exterior buffer. But in the meantime, it's also highly membrane permeable. So it will permeate through the lipid bilayer and then interact with the internal gradient. In this case, if you put a trapping agent inside, it will form complexes with this trapping agent and get locked inside the liposomes. So this is the standard active loading technology. but it provides several advantages. For example, the drug loading capacity and efficiency is much higher with active loading compared to the other method called passive loading. And because the drug is stably loaded inside, the in vivo stability is increased. So these are the two major advantages. But the limitation is that this technology only applies for amphipathic drugs, meaning the drug needs to have good solubility, a good membrane permeability to be able to actively load it into liposomes by this technology. But as we know that most of the investigational drugs are poorly water soluble. So that's what we are focused on, developing this so-called solvent-assisted active loading technology, in short, SALT, for poorly water soluble drugs. So again, our idea is that first, we still create a liposome formulation containing a transmembrane gradient. But before we add our drug, we actually included a small amount of water-miscible solvent in this liposome formulation. And when we add this poorly water-soluble, then the solvent will have two functions. First, it will increase the solubility of the drug in the mixture. Second, it will increase the membrane permeability, allowing the drug to penetrate into the inner aqueous core to form complexes with the trapping agent. At the end, we just perform dialysis to remove the solvent or any unencapsulated drug to obtain the final purified liposomal drug formulation. So that was just a concept. So the first thing we did to demonstrate this is a feasible method is using a model drug to test our hypothesis. So the drug is called staurosporine with a structure shown here. Although it's a weak-based drug, the drug is actually not soluble in water. It's only soluble in DMSO. So our approach is that we create this ammonium sulfate gradient for weak-based loading. And then we compare either no DMSO in the loading mixture or the loading mixture contains 5% DMSO, and we compare the drug loading efficiency. As you can see, without DMSO, all the drug is precipitated outside the exterior phase without significant drug loading. But with 5%, we were able to load all the drug molecules inside the liposomes. And so the cryo-TM, just to demonstrate that indeed the drug is loaded inside the aqueous core rather than the bilayer, like most other reports have demonstrated. So as you can see that you can see drug crystals inside the PFSUV particles, liposomal particles. Okay, let's look into the applications of this SALT technology. So the first example is a Chinese medicine. So as you know that a lot of active compounds have been isolated from traditional Chinese medicine or herbal medicine. But they do have significant problems. First, most of them are actually not water soluble. So one example is gambogic acid. The structure is shown here is a weak acid drug, but most of the structure is very hydrophobic. So as a result, gambogic acid is not soluble in water. But it has been shown to have several really exciting biological functions or pharmacological activities. So including anti-inflammatory, anti-angiogenic, angiogenesis and anti-proliferation. So this drug has been heavily investigated for cancer therapy. But again, the water solubility is one of the limitations. So in here, we are using this copper acetate as a trapping agent for this model drug. And we were able to show that we could use many different solvents in the loading mixture, containing just 5 to 7% water miscible solvents of this list of solvents to actively load gammatic acid into the liposomes. So I just want to show you that this is our final product of Lipo-GA. The size is about 18 nanometer, and the drug to lipid ratio is 1:5 weight ratio, so very high drug loading. And as I mentioned, the problem with gammatic acid is poor solubility. So we were able to increase the apparent solubility from 8 microgram per ml to greater than 10 milligram per ml. So very significant improvement in terms of solubility. So what is the working mechanism for salt? So what we have done is a very systemic analysis between the loading efficiency and the percentage of miscible solvent within the loading mixture. And what we have observed is that there is definitely a optimal solvent range for complete drug loading. Okay, so below that is not good, but exceed that range is definitely not good either. And another thing is that we also discovered that if we want to achieve complete drug loading, we need to add a little bit more solvent than the amount that is required to completely dissolve the drug. So the results suggested that the solvent is acting through two different mechanisms. First, definitely to dissolve the drug, but just completely dissolving the drug is not enough, because you need slightly above that. So we are anticipating that maybe the slightly over solvent requirement is to increase the membrane permeability to allow the drug to penetrate through the bilayer more effectively. And also, if you include too much solvent, then the membrane might become unstable and the gradient might collapse to prevent drug loading. So this is the working mechanism for salt. Let's go back to the gammatic acid story. So poor water solubility is one of the limitations. Another limitation is that GA induces significant hemolytic toxicity. So in here, we are comparing the hemolysis of free GA with our Lipo-GA. As you can see, free GA is very toxic to RBC. But after liposomal encapsulation, the drug becomes safe. So we also look into the anti-tumor efficacy. We look at multiple animal model or tumor models. Maybe I will just focus on the EMT6 breast cancer on the left-hand side. As you can see, the tumor is quite aggressive. They grow very fast. With the free GA treatment shown in green line here, shown in the orange line here, there's no significant effect. Probably because of the dose we could safely inject it was not effective. But if we load the drug into liposomes, even at a low dose, we were able to show significant results. And because of the reduced toxicity, we could even inject a much higher dose at 20 milligram per kilogram, and we were able to completely completely suppress the tumor growth. So this is another model showing comparable results. And I also want to introduce a second application, which is quite interesting in my opinion, that we use this technology to help us prepare child-friendly oral formulation. As you know, that pediatric formulation is not a lot of people working on this field because first, it's very challenging. Second, the market is small. But I think this is a very important topic to work on, especially for academic scientists. So what are the requirements for pediatric formulations though? First, liquid over solid, because most children have a swallowing problem. And the taste is very important. And only safe ingredients and bioavailable. And I was thinking, well, maybe our liposomal formulation will serve the purpose, right? Because we could load drug inside the particles, and we could, in that way, we could increase the solubility to prepare a liquid formulation. And also, the drug molecule is now loaded inside the bilayer, and the bilayer will prevent direct contact between the drug molecules with the taste buds. And this would be an effective way to mask the taste of many drugs. And again, I'm using malaria drug as a model drug. So I'm going to skip the introduction, just go into the results. So the model drug is mefloquine. It's super bitter. It's still the first line drug for malaria in Africa, but it doesn't really have an pediatric formulation. So in those countries, what has been done is that pharmacists will just take adult tablets of mefloquine and break it into pieces and crushed and into powder, and then add to water or liquid or food and feed to the children. But as you know, that is not going to mask the taste well, so the medication adherence is not good. So many children just spit out. That affected the accurate dosing and the medication adherence. So we use our technology, the salt technology, to load mefloquine, which is a poorly water-soluble drug, as well into the liposomes. And the first thing we look at is the taste. So we measure the bitterness by the e-tongue technology, basically a biosensor that is able to quantify bitterness. And as you see here, we use 10% sucrose as a reference. It shows negative results, suggesting it's sweet. And mefloquine suspension, as you know, is right now used in Africa, very bitter. And we have infant Tylenol as a reference too, just to show that what is the acceptable range of bitterness, which in this case is 250. But look at mefloquine liposome, it tastes like a sugar water here. The next thing we look at is looking at whether our formulation can release mefloquine in simulated gastrointestinal fluids. And here is simulated saliva. We don't want the drug to release in the saliva. So again, no drug release. And in here we have mefloquine suspension, very rapid release, so causing bitterness in the mouth. And we have two mefloquine liposome formulation. The only difference is just a different drug to lipid ratio. So you see no drug release. But in the simulated gastric fluid, you see rapid release for all formulations. And in the simulated intestinal fluid, we see rapid release as well. So the data suggests that our mefloquine liposomal formulation will not release in the cavity to induce stiffness, but will be rapidly released in the gastrointestinal conditions to allow the rapid drug absorption. And finally, we look at the oral absorption in a mouse model. So just very quickly, you see that our liposomal mefloquine shown in purple line here is absorbed very rapidly, producing a very good blood profile in mice. It's comparable as mefloquine suspension, or even a little bit better because it increases solubility. So another example I want to show is lyophilized mefloquine liposome. So we know that liquid formulation is difficult to store. So the lyophilized mefloquine liposomal formulation is to resolve that challenge by drying the liquid formulation into powder. But this powder formulation is able to be reconstituted in just a second. And we show that with this lyophilized formulation, we were still able to produce very good blood profile. And we also confirmed that after lyophilization, mefloquine drug molecules is still loaded inside the liposomes. And the final part of my talk today is, again, using this salt technology as to prepare a immunotherapy formulation for treating intraperitoneal metastasis of cancer. So we know that intraperitoneal metastases are often found with ovarian cancer, pancreatic cancer, and colon cancer. They are very difficult to treat. And one of the characteristics of these metastases is that the tumor microenvironment is highly immunosuppressed. So we were wondering whether we could use a drug delivery technology to wake up the immune system in the peritoneum cavity to promote anti-tumor immunity. So our approach is that we want to use an immune boosting agent, and we want to localize this immune boosting agent in the peritoneal cavity. So the immune boosting agent we are using is RA4A, is a toll-like receptor 7 and 8 agonist. So it's very effective in activating dendritic cells to secrete interferon alpha to promote anti-tumor immunity. But the problem with RA4A is, first, is the water is truly water soluble. Second is that if it's injected and distributed throughout the body, then it will induce non-specific immune response globally or systemically to induce very significant side effects. So the delivery must be localized to the peritoneal cavity to reduce the toxicity and improve the efficacy. So again, we were thinking to use our salt technology to load these drugs into liposomes and use liposome to localize this drug in the peritoneal cavity. The first thing we did was looking at how different formulation of liposomes would affect the peritoneal retention. We compare the charge effect. So we prepare negative, neutral, and positive formulations of liposomes, and then IP deliver these formulations into mice and look at their IP retention. As you can see that the positively charged liposomes are very effective in retaining in the peritoneum compared to the negative and neutral liposomes. So especially for negative formulations, you see a very rapid absorption after 1 hour and to the rest of the body. So then we quickly look at the pharmacokinetics and the biodistribution of the formulation in comparison with free drug. in these components, including peritoneal fluid, IP cells, which is cells collected from peritoneal fluid, and then plasma. So what we hope to achieve is that liposomes or DSTAP-liposomes was able to increase the peritoneal fluid concentration, increase the delivery to IP cells, but reduce the absorption or slower absorption into plasma. And you see that we achieve these as shown that the red data representing DSTAP-RA4A are much higher in the peritoneal fluid and IP cells, but the absorption to the systemic, to the system is much delayed compared to the free RA4A. And we were also wondering, within those IP cells, what kind of cells are we deliver our DSTAP-RA4A to? So we labeled the DSTAP-liposomes with a fluorescent dye and IP injected into mice and examine those Di-positive or Di-labeled liposomes and Di-positive cell population. So within all the Di-positive cells in the IP, in the peritoneal cavity, a lot of them, like 50% of them are monocytes. And about 10% are dendritic cells, 10% are NK cells. So this is to our expectation, okay? But we also look at within each cell population, how many of them are positive with the Di. For example, monocytes here. Actually, among all the monocytes in the peritoneal cavity, only 0.5% are positive. So actually, in this, we can conclude that monocytes are not selectively targeted by our formulation because only 0.5% of them are positive. The reason that we see a lot of Di-positive cells are monocytes is because monocytes are the vast majority population in the peritoneal cavity. But what is really interesting is the dendritic cells. We see very, very high percentage of the dendritic cells are positive with Di, about 40%. And this mature macrophages are very effective, are very effectively targeted as well, but their population in the peritoneal cavity is pretty small. So the results suggest that the major immune cell population that we are targeting to is dendritic cells, which is what we hope to target. As I mentioned, interferon alpha is the major cytokine induced by the RA4A activation, and also the very important cytokine that will turn a cold tumor into a hot tumor. So this is the cytokine that we look at first. So as you can see here, we look at the mRNA expression from these IP cells, and you see that with DSTAP treatment, we were able to increase the interferon alpha expression by 10-fold compared to the control, while free RA4A was less effective, only inducing 5-fold. And finally, we look at the efficacy because all the data are very positive in supporting that we are inducing a very good immune response. So in here, we are introducing CD26 colon cancer into mice by IP injection. We also label these cells with a luciferase tag so that we can monitor the tumor progression non-invasively using bioluminescence imaging. And on day one, we inject oxaliplatin, which is commonly used anti-cancer drug for colon cancer. And the next day, we IP inject either PBS, free IFOA, or DSTAP IFOA. And then we monitor tumor progression using bioluminescence imaging. As you see that these tumors actually grow really fast. So on day 13, the tumor burden are really significant. But with oxaliplatin DSTAP combination, we were able to slow down the progression. And here we show the quantitative results. And in here, the white bar shows oxaliplatin plus PBS. There's no control of the tumor progression. With combination of free IFOA, you see initial response on day 3 and day 4, but tumor quickly rebound and uncontrolled. And so, as I have shown earlier, free IFOA was able to induce interferon alpha release. So, this early effect probably from non-specific effect from interferon alpha. So with the combination with DSTAP, the effect is more prolonged. Even on day 13, it shows significant tumor inhibition compared to PBS and the free IFOA group. So our data suggests that there might be some immune response against the tumor. So another thing we look at is whether our treatment has changed the immune microenvironment. So in here, we isolate the peritoneal fluid and then look at the T cell and differentiate them between the CD4 and CD8 population. So in the first group is oxaliplatin plus PBS. You see that the CD8 population is 76% among all the T cells, and CD4 population is about 16%. And after combination with RA4A, you see the CD8 population increases to 82%, and CD4 population decreases to 10%. With DSTAP RA4A treatments, the CD8 positive population further increased to 93%, and CD4 population further decreased to 3%. So for the anti-cancer therapy, CD8 population is preferred because they are cytotoxic T cells. They were able to recognize cancer cells and then perform lysis. So we believe the mechanism is preferred for significant cytotoxic anti-cancer effect from the immune response. So this slide just summarizes our proposed mechanism supported by our data. We believe that oxaliplatin is first induced double-strand DNA breaks, inducing so-called immunogenic cell death, and the released antigen together with DSTAP RA4A delivery to the DCs will activate the dendritic cells to the active part, to the active counterpart, and then the activated DCs will secrete interferon alpha, and then they will interact with T cells and then activate T cells into CD8 positive T cells. And these CD8 cells will then perform tumor lysis to suppress the tumor. So I just want to quickly summarize my talk today. So again, we focused on formulation and the manufacturing method development for liposomal drug delivery systems. And we use these technologies to target drugs to specific sites. And today I give you one example for hepatocyte targeting. And second, we are interested in drug loading technologies, especially for poorly water-soluble drugs. And today I show you several examples of how we can use this solvent-assisted active loading technology to load a variety of drugs to improve their targeting. I just want to acknowledge my lab and my collaborators and funding. Thank you so much for listening.
So thank you, Professor Li. So we can have a couple of quick questions if anybody have.
Thank you, Deepak. I might ask a question.
Yes, Professor.
Professor Li, very nice presentation. I learned a lot. I had a question about your the salt loading technology. When you're using solvents like DMSO or DMF, how difficult it is to remove them after you formulate them? And was it done by dialysis or freeze drying? How did you do that?
Yeah, so far we were using dialysis to remove those residual solvents, and we monitor residual solvents throughout the dialysis process. And what we found is that after 2 hours, those solvents are under detection limits. But we actually performed dialysis much longer than 2 hours. And we did multiple changes of the solvent, I mean the buffer.
Sure. And then the other quick question was that these lipids that you're using, are they able to survive the lipases and the gastric environment? Are they lipids that are specifically chosen for oral delivery?
Yeah, so I think you were referring to the pediatric formulation part. So we did see some very rapid release in the simulated intestinal fluid. And so we actually examined the mechanism. So first we were wondering whether it's through the lipase digestion, but it's actually not, because when we incubate our liposomes with the lipase that we purchased from Sigma, and we incubate them for several hours, and we didn't see any degradation. So the release mechanism is actually through the sodium tauglate or the biosalt that we supplemented into the simulated fluid. Because in the normal intestinal conditions, there will be some biosalts presenting in the intestinal fluid. So that's what we have found to be the mechanism.
Okay, excellent. Thank you so much.
Yeah.
Sir, there is one question. Like you have talked about the amphiphilic drugs. So what are the properties or the characterization we can do for these drugs, and how the nanocarriers should have some property or groups on it so that it can be loaded efficiently inside the formulation?
Yeah, so if you are talking about the salt technology for loading drugs into the aqueous score of liposomes, we do have some criteria for selecting those drugs. Definitely, you need to have a good loading gradient and so-called trapping agent inside the liposomes to interact with your drug, right? So the most successful example we have obtained so far are wheat-based drugs that containing amino groups that can form complexes with sulfate ion. But we were also successful with the carboxylate functional groups, and we would use copper gradient or copper to complex with those functional groups. So I think the functional groups presenting in the drug you are hoping to load is very important. That will decide what kind of gradient you need to use. But in terms of our limitations, because our technology is trying to resolve the issue of or the limitation of current active loading technology that require drug to be both soluble in water and membrane permeable, right? So I think I have shown you several examples that as long as the drug is, has some functional groups that you can utilize,
Yes.
There's a high chance that our active loading technology will work for them.
Okay. Sir, another small question is, if the drug is low water soluble and low low lipophilicity is there, then what will be the liposomal fate according to the formulation?
Right. So I would say most of those are low solubility and poor membrane permeability drugs. They are because of they are a substrate for PGP, right? So that they are characterized in that category. But in our liposomal delivery, because there's no PGP there, so normally if the chemical structure shows that the permeability is okay, then it should be able to load.
Okay, thank you, sir. Thank you.
Yep.
Yeah, thank you, Professor Li, for providing the insights on the phospholipid-free SUVs for targeting and discussing the active loading technology with the audience. So I thank you for sparing your time for this talk. Thank you, Professor Li.
Thank you. Yeah, thank you.